Okay, does anyone have questions for Donna?
Carol's question is, can you explain an application to a specific research project, like you had a slide about the warm season grass?
Well, you may want, let's see. Well, let's say that you want to find out what would, this is not the warm season grass, but you may want to see how quickly warm season grasses can have an effect on soil microbes. Or here's another one. Let's say that you want to find out what's the optimal amount of phosphorus to add with the, I mean, if you add a certain amount of phosphorus, you can actually decrease the amount of mycorrhiza because there's so much there that the plants don't need to work at it. So you may want to find out what happens to mycorrhizal levels when you add different amounts of phosphorus. You may want to look at different kinds of cover crops. You may want to look at adding manure. You may want to see what happens with different kinds of herbicides. Any kind of treatment that you can add to soil, you can have a control and a treatment and compare the phospholipid fatty acid numbers. You can tell both which ones have the most microbial biomass, and you can see which of the soil, which of the groupings are affected most by it also. So, I mean, there's just any number of treatments and you can see the effects. Or maybe you want to increase your mycorrhiza. You could look at what could I do to increase the mycorrhiza or the gram-negative bacteria. Or maybe you want to look at it from the other point of view. Where do you have really good other soil health factors and maybe really good yields? What are the microbial characteristics of that soil? Does that help?
Okay, we have another question from Les. What management decisions have been made based on the PFLA analysis?
I don't know that the management decisions, I think we're more in the part now where we're trying to see, okay, if we make this management decision, how does that affect the soil microbes? So I'm not sure that we're at the point where we take it, run a sample and say, well, this soil is really low on this. What could we do to increase mycorrhiza or gram-negative bacteria or total numbers? Well, I think I do have one client that they have, I think, a consulting firm. So they do soil analysis and plant tissue analysis. So they send us samples, and they've sent probably 150 samples. So they are making management decisions on that. And they've found a lot of correlation between the soil chemistry, and sometimes they've been able to diagnose plant pathogens, like if they send in a poor bean soil sample and a good bean soil sample, and you have a lot of fungi in the poor bean, you may have a fungal pathogen. And they may have also found that it's just overall not a lot of microbes in there, so they can add manure, or they can figure out what the farmer wants to do to provide more food and habitat for the microbes.
Very good, Donna. Todd has a question. Could PLFA be used to replace in-field biomass collection with plant mass weighing above and below ground?
I don't believe so. I mean, I guess you could. Because with the PLFA, it's just the microbial biomass. So you're not getting root biomass, you're not getting above ground biomass. I mean, I guess you could say that, you know, the more plant biomass you're adding, the more microbes you would be getting, theoretically. So I guess you'd have to look at the correlation. Maybe you could correlate the below ground biomass and the above ground biomass with the level of microbes that you have.